Oh, so now do you want headphones? Okay, cool. All right, team. Ready? Give me another second.
I need to go.
Need to move? All right, cool. So, just pointing out basically our to-do list for microbes. Today, so there's 3 things I still want to cover with you guys as clarifications, just to make sure you guys understand what kind of the issues were with some of your internals. I have decided to break it up so that way you guys have some self-directed study time so you can do those corrections, or you just basically get a break from me talking, but I'll see how you guys go and you guys can decide whether or not you want me to keep going and just get it all done at once, or if you want to stop and have some time to work. Basically the plan is I'm going to give you guys another week to do this internal because we have either some extra notes I want to give or you guys need some processing time. So there are 3 things that I commonly saw as an issue with the reports that were handed in so far. So I want to talk about each one of those points and give you guys some examples. I also do want to talk about an example. I also want to talk about what I mean by these different requirements using cancer as the exemplar, because you can't use cancer to write up. So the thing that I saw, so everyone that's handed in microbes so far, the highest grade I've given is an achieved, so don't feel bad if you've gotten an achieved, because the big issue that people had was this requirement over here, which is explain the abiotic or biotic factor of the human body and how it interacts with the life process of the pathogen. A lot of you guys had the information there, but didn't unpack it and explain why. And I realized in hindsight that you guys weren't connecting stuff from the previous lessons on the microbes unit. So let me explain what I mean by that, and then I'll point back to these various lessons and how that integrates. So, and I'm just going to try to find my notes from my notebook on stuff that I saw as common things. Microbes, there it is. Cool. All right, so I made some notes here for me to remember, but I'm showing it just so you guys are aware of some of the things I noticed. So a lot of you guys were able to identify abiotic or biotic factors that affect the pathogen's life cycle. For example, temperature was probably the most common one I saw, but you didn't explain why temperature was a factor in the life process. So I was very mindful you guys need a lesson on that. I also had some other things to think about, like immunocompromise, moisture, nutrients, that sort of stuff. So let's talk about temperature and why that actually is a factor for our life processes. The first thing I want to talk about, though, I'm just looking for my images, and hopefully I still have all of them because I think some of them fell out of my bag. I'm just trying to decide which one I want. No, I'm going to write notes first. Okay. I think we did this in a lesson, but I can't remember which one, so I'll just recap it. So something to keep in mind when we are talking about reactions and chemical reactions is that there's certain factors that need to happen. So for a chemical reaction to occur, there are certain requirements that need to be met in order for that reaction to happen. And when we are talking about things and their life processes, living things are going through a wide variety of chemical reactions in order to survive. Even the viruses are doing a sort of, actually, yeah, they're doing a chemical reaction because they are trying to make viral replications of their DNA or RNA. So you see this all, like in all living things. So, first thing in order for a chemical reaction to occur is that the reagents need to meet. If they do not meet together, there's no reaction. Just be mindful when we talk about reactants. Reactants are what become products. So if we think about equations, They look like that. The common one that we were talking about as one of our life processes is respiration. So let's say you have sugar plus oxygen, which I think is 6. And that burns to become it becomes carbon dioxide in water. So that is a chemical reaction there. These here are my reactants, these are my products. And when I do that, I produce energy. So we always have to have chemical reactions in our living things. Now, in order for that reaction to occur, the things need to meet. So the oxygen needs to meet the glucose molecule, the sugar molecule. If it doesn't meet, there's no reaction. Are we okay with that so far? Okay? The second thing it's going to need is enough energy. So whenever chemical reactions occur, it needs some amount of energy to get it going. So it needs energy. And I should say it needs enough energy. You can have energy, but if it's not enough, the reaction won't occur. Just make sure I'm spelling the next word right. The last requirement it needs is that it needs to have the right orientation. And so for example, let's say I have this as one of my reactants and my fist is the other reactant. In order for the reaction to occur, these two things need to be in the right orientation like this. If my hand's like that and these two particles meet and they're not in the right orientation, they're not going to react. Does that make sense? So needs to meet with enough energy, needs to meet in the right orientation. So those are my requirements. Now, what I can do is I can influence this rate of reaction. I can increase it or I can decrease the rate. So let's talk about ways that's going to be influenced. And I'm just trying to find which pictures I want. I remember. I would definitely want this one. And I want this one. You gotta find the ones I'm looking for. That one. And I want that one. I'll leave that one till later. Okay. So, I think I wrote down which order I wanted to give these to you guys. Let me see where it goes. Oh, just temperature and enzymes are last. Okay. Cool. So, here are some things that are going to affect the rate of reaction. The first thing is concentration. So, I'm using this one here. So, this is the first thing that affects reaction is concentration. Let me find a color that's different. Okay. So, this on the bottom graph, I'm just good idea to kind of refresh how do we unpack what graphs mean or different data means. So this here is the time from the start of the reaction. So here 0, the reaction hasn't started yet, and as time progresses, the reaction continues. What you notice over here is something called volume of gas. The gas is one of the products. So we can measure the gas amount to know how fast the reaction is going. So when we look at high concentration versus low concentration, which one produces gas faster? The high, because we can see that it's making more gas that way. So what this tells me as a first step is that concentration will affect the rate of the reaction. If I increase the concentration, the reaction goes faster, and if I decrease it, it goes slower. The reason why is when I have a more concentrated solution, I have more particles, and since I have more particles, they're more likely to meet. So colliding is basically what we're talking about when we say meet. Say the word collide is the science word we use. Are we okay with that so far? Yeah? Do you guys notice how it starts off really, really steep and then slows down with time? So the volume of gas is being produced in a slower manner? Why is that? Any idea? Has to do with concentration. So the reason why this slows down with time is because we're using up the reactants. So the reaction's slowing down. So rate of reaction slows as reaction progresses. Because reactants are being used up. And more products are forming. So the downstream effect is that then they are less likely to collide. Sorry. Hopefully you guys are okay. Do you also see how these two reach the same endpoint? So both of the reactions go to completion. It's just this one reaches the endpoint sooner. Okay? So we still get the same endpoint, but the first reaction with the higher concentration happens faster. So that's the first way we can manipulate our reactions. Questions, concerns, comments? All right, ready for the next one? Next one, looking at this one. All right, the next way we can influence our reactions is by surface area. So these three substances all have the same volume. There's the same amount of matter there. But what I've done is we've chopped up the matter differently. So if we chop it up into smaller pieces, it exposes more of the surface, it increases the surface to volume ratio, and therefore there's more likely to have collisions. I'm just going to start abbreviating stuff because I don't think you guys want to write that much either. ROR stands for rate of reaction, and then these three dots mean therefore. That's a math term. So the example that I have is, you know how you have the cubed sugar, like the ones that come in like the square that's already like in a little, yeah, and then you also have sugar that's in like teaspoons and stuff. It's obviously going to be easier to dissolve the little teaspoon of sugar that's loose instead of the cubed sugar that you just drop in there. And that's because it has more surface area. And I think we did a practical with this. I'll have to double check. I can't remember what we did for term one.
Nice.
Yes.
The word surface to volume.
Yeah, surface to volume ratio. I'll try to write my next one neater. Yeah. With the agar jelly? Yeah, so you have seen this. Yeah. Yeah, we have done this. That was a cool experiment. Remember you guys wanted to eat it, and I was like, don't eat it. It's a good way to end up in the hospital. Yeah. With the vinegar? All right. Here's our next one. That's the next one I've chosen. All right, this one here is temperature. So we notice at a high temperature versus a low temperature. So which one causes the reaction to go faster? The higher temperature, yeah? So this relates into a lot of the data you guys were talking about, because you were talking about how a lot of these pathogens want to live at our temperature. And a lot of the benefit of living at our temperature is that it's going to help with these rates of reaction. It's going to make these reactions happen faster. The reason why the higher temperature has a faster rate of reaction has to do with two factors now. First factor is my particles are more likely to collide because they are moving faster. Sorry, are moving faster. Therefore, more likely to collide. And then the second reason is that the heat from the increase in temperature can be used as activation energy. So now we have more energy. Which is one of the requirements. You need to have enough energy in order for that reaction to occur. So that's why temperature is a big factor and why things want to be at certain temperatures. Are we good so far? Again, in both these examples, you're getting to the same endpoint, but this is going to get to the endpoint faster. Something else to keep in mind, or as an example, if you guys aren't still kind of waffy on that, think about like when you're trying to make raro and you're trying to dissolve the sugar into cold water versus when you're trying to make a like hot Milo and you're dissolving the Milo in the hot water. It's a lot easier to dissolve the Milo than the raro because of the temperature of the water. All right. Next one is this table here. Not table, graph. We should get blue. Okay. So last one I need to talk about is enzymes. So just as a reminder, when we talk about enzymes, we are talking about a protein that can carry out chemical reactions. Oh my God, what's going on with my brain? It's important to remember that enzymes are not a reactant, so they do not get used up in the reaction. Now, what these enzymes do, so you can see here, when I have an enzyme, it's a very fast reaction, but with no enzyme, it's very, very slow. So the enzymes make a huge difference. What enzymes do is, let's say I have this substrate that has that shape, and I have this reactant with that shape. What an enzyme does is it basically acts like a matchmaker. And it helps my reagents meet in the right orientation. And when it does that, it lowers the energy that you need for the reaction to occur. So it lowers activation energy. and it helps the reagents get into the right orientation. Or reactants, I should say. All right, keep going. Weird size. I should have probably cut this. I'll grab my scissors since you guys are ready. Good so far? Any questions? Okay. This is the next one I want. Actually, I'll put this more centered. There we go. That's the last image. So this one here. All right. Something else that is important to remember when we are talking about temperature, this is actually linking temperature and enzymes together, I should say. And you guys saw this. You guys saw this on the whatchamacallit. You saw this on the exemplar that was put online. So this is talking about rate of reaction. This is temperature. And what you'll notice is that the reaction obviously increases with the temperature increasing. Are we okay with that so far?
Yeah.
Which makes sense because it matches what we saw over here, right?
Yeah.
But there's a problem. Once you hit above, it looks like 48 degrees, the rate of reaction decreases quite suddenly. See how it drops? So this is referring to the optimal temperature of enzymes and what they like to operate at. So you see how it says optimal rate. That basically is telling you the ideal temperature. So what happens after this point? So the good news, like up to that point, it's really, really good. The particles are moving faster. There's more energy. They're more likely to collide. Our rate of reaction is going really, really well. I eventually hit a roadblock because now I've gone too hot. And when I've gone too hot, what happens is denaturing. And so the enzyme breaks apart. So temp gets too hot. And the enzyme breaks. Actually, the enzyme, I should say denature, which is breaks apart. So you have to remember this enzyme has a very specific structure, and what happens is it basically just starts to unravel. And if it's unraveled, it doesn't work. So that is why there is a very optimal temperature for where these pathogens want to live. And our body's really good because it gets us to that point for them without going over. If it gets hotter, then it goes over, it stops working. And that's part of what the body's doing when the immune system reacts. So when you notice when you get sick, your body temperature increases, and it's increasing because it's trying to do things to kill the pathogen, like push it over the edge. Now, it's not good for our cells either. So when we have an immune response, we don't want it to be the nonstop reaction because we don't want to be ill the whole time. It's not actually good for our cells. It's just a temporary solution to get rid of the pathogens. All right. Questions, concerns, comments so far? No? All right. So keep that in mind when you guys are writing for that merit bit, is that you pick one of those factors and you have to then unpack why it is going to benefit. So going back to that list that I had. So temperature, a lot of you guys picked that one, which makes sense. Be really careful when we're talking about temperature that you are not talking about temperature in a sense of the outside temperature. So when you're talking about winter and why we have more viral spread in winter, the cold weather isn't the ideal temperature for the pathogen. It's because people are indoors. It makes it easier for the virus to move. Okay? So keep that in mind, that there's two different temperatures you're thinking about. You're thinking about your body temperature. You're also thinking about the temperature of the environment. And more likely, the one you need to be writing about is the temperature of our body. Moisture is an important thing that some of you guys mentioned, the need for water. So be mindful that we are water-based organisms, and so that provides that moisture. A lot of these, not so much the viruses, because the viruses are not living, but definitely for a lot of the molds and the bacteria, they want to be in a water environment because their cells would then need to maintain their like homeostasis. So their cells want to have the right amount of water too, so they don't burst open or they don't like shrink super tiny. And then nutrients is something else that our body provides a lot of because we're basically a food source. We eat food. Our gut has a lot of food in there. Our body has a lot of food. Our body also has a lot of oxygen because we are aerobic creatures. So lots of resources in our body. Okay, so that's what you need to think about, and that's what you need to be explaining. I'm just mindful of time. I've been talking for about a half hour. I might leave the other two bits of data for Monday. Or sorry, Tuesday. Does that sound good? I'm thinking you guys had enough too? Okay. I'll leave the last bit and we'll just keep going. So Monday we'll do notes. Tuesday we'll do notes. Thursday we'll do notes. And then Friday I'll just let you guys have a study day. Okay? And then we'll see how you guys feel if you're ready to hand in at the end of next week or if you need some more time. We probably won't do class time anymore, but I'm happy for you guys to work on it outside of class. So we'll see how you guys go. Okay? All right. That's us.